Now continuing with the next page. Penny types are bilaterally symmetrical and centric types are radially symmetrical. Right? And diatoms have cell walls containing silica, constructed in two overlapping halves which fit together like two parts of a soap box. Penny types or pair of petri tissue centric types. The outer wall is called epitheca and the inner wall is called hypotheca. They are present in both fresh and salt water and comprise an important food for the aquatic animals. However, out of 5,500 species, most are marine. They lack flagella and float mainly due to light storage, lipids present in them due to their silica impregnations. The walls of diatoms are indestructible. As a result, diatomaceous earth formation has occurred due to remains of cell walls of diatoms in the forms of fossils. Silica wall of diatoms is designed with fine ridges, lines, pores, etc. These ornamentations are either radially symmetrical or bilaterally symmetrical on either side of the long axis of the cell. Food reserves is oil and chrysolaminarin or leucosin beta 1 to 3 glucan. Excuse me. Multiplication is through binary fission. Binary fission reduces the size of most daughters due to relation of one valve of the parents. This is corrected through the development of rejuvenescent cells or oxyspores. For example, Cocinodiscus, Stephanodiscus, and Cymbella, etc. Diatoms may exhibit slow gliding movements produced by streaming of cytoplasm through grooves on the cell wall. Due to various designs over the cell wall, they have been regarded as jewels of the plant kingdom. The oil is extracted from some fishes and whales, especially the ones produced by diatoms. Diatomite deposits are often accompanied by petroleum fields. Much of the petroleum of today is probably due to decayed bodies of the past diatoms. Diatomite is porous and chemically inert. It is therefore used in filtration of sugars, alcohols, and antibiotics. Diatomite is employed as a cleaning agent in tooth paste and metal polishes. Diatomite is added to paints for enhancing night visibility. Diatomite is employed as insulation material in refrigerators, boilers, and furnaces. Diatomaceous earth is added to make soundproof rooms. It is a good industrial catalyst and a source of water glass or sodium silicate. Diatomite was at one time employed in handling the storage of dynamite and strong acids. Dinoflagellates. They comprise the phylum Paraphyta of algae, golden brown photosynthetic protists. They are single-celled. Most of them are surrounded by a shell made up of thick interlocking plates covered with cellulose cell wall. They are motile with two flagella, hence dinoflagellates, one projecting from one end and the other running in a transverse groove. Like diatoms, they have a phycoxanthin in addition to chlorophyll. They are autotrophic and photosynthetic. For example, Serratium blastodium is a colorless parasite on animals. The food reserves is in the form of oils and polysaccharides. Most of the dinoflagellates are marine and important photosynthesizer in the ocean. Occasionally, members like Goniolax accumulate in large numbers in some parts of sea, coloring the sea water red and are responsible for red tide. Goniolax causes red tides because it shows bioluminescence of phosphorescence and makes the sea look red. Some species of dinoflagellates are poisonous to vertebrates, and when these accumulate, large number of fish in the region of the ocean may be killed. A non-contractile vacuole or passule is present due to presence of two flagella at right angles to each other. The dinoflagellates show peculiar spinning movements, hence they are called whirling whips. Nucleus is mesokaryon with condensed chromosomes, even in interphase. Histone is absent. Division occurs through denomitosis, in which the nuclear envelope persists. Microtubular spindle is not formed. Chromosomes are asymmetric and move while attached to inner membranes of nuclear envelope. Dinoflagellates may have eye spots, trichocyst, example peridinium, and nidoblast, example nematodinium. Right to the next page. Some forms show bioluminescence or phosphorescence and are called fire algae, example Noctiluca goniolix. Method of reproduction is only asexual. Sexual reproduction is usually absent, exceptional serration. Some species of dinoflagellates are taken as food by mussels. These mussels remain unarmed by dinoflagellates, but if man eats these infected mussels, he may fall ill. Euglenoids. Euglenoids includes flagellate protists of freshwater and damp soils. Presence of anterior invaginations like some ciliates has given the name plant animals. Euglenoids are more advanced than blue-green algae from evolutionary point of view, but they have a definite, easily stained nucleus, and the chlorophyll is not scattered in granules, but is localized in chloroplasts as in higher plants. The nuclear envelope persists during division. They are free-living, found in freshwater ponds and ditches or in the damp soil. Euglenoids are characterized by absence of subwall, but they do not contain flexible pellicle made of proteins. All the euglenoids have one or two flagella, tinsel and dactylum, by means of which they can swim easily. Euglena is more readily available protist for laboratory studies. Euglena bears a flagellum inserted at the anterior end in a cavity. They bear a red pigmented eye spot and a gullet near the base of the flagellum. The pigmented eye spot is astaxanthin. A swelling called paraflagellate, paraflagellar body is found in the region of union of flagellar roots. Photosynthetic form bears many radiating chloroplasts of various shapes. The chloroplasts contain pigment like chlorophyll A, chlorophyll B, xanthophyll. Nucleus with one or more prominent nucleoli occurs. Pyrenoids may or may not be present. Some euglenoids are green and holophytic, photoautotropic like other plants. Few are non-green and saprobic like fungi and bacteria. Some capture and ingest the organisms like animals. Holotropic. Green forms have saprobic mode, pick up organic matter from outside. Mixotropic. Holotropic or phagotropic nutrition is absent in Euglena. Euglenoids store carbohydrates in the form of paramecium chemically, distinct from starch and glycogen. Reproduction is usually asexual by cell division, but sexual reproduction has been reported in one genus. Under favorable condition, euglenoids were produced by simple longitudinal binary fission. Flagellum disappears prior to divisions. Most of the species produce cysts, have a thick stratified membranes with deep red coloration due to synthesis of hematochrome. Besides cyst formation, many of non-flagellate cells may get embedded in a common gelatinous sheath resembling a palmella stage as in algae, example Euglena, Paranema, Cephalomonas. Slime molds. Slime molds are consumed or decomposed of protists. Asexual reproduction takes place through binary fission, plasmatomy, spores, cyst, and sclerotium. Sexual reproduction is isogamous or anisogamous. Both zygotic meiosis, cellular slime molds, and gametic meiosis, acellular slime molds, occur. Slime molds have characters of plants, cellulose cell wall, animals, phagotropic nutrition, and fungi spores. They take part in both decomposition of organic matter and feeding of other decomposer organisms. Slime molds therefore live in contact with organic matter. Slime molds 50 sp have several features which are animal-like in their vegetative stages and plant-like in reproductive stages. These slime molds are widely distributed, growing in damp and shady places. They may be found in the soil rich in humus, damp old planks of wood, rotting logs, decaying leaves, etc. They prefer moisture and darkness or dim light for normal growth. They move to drier and exposed habitats during reproductive phase. More than 100 species have been reported from India. The vegetative phase of thallus is a free living type, which is naked multinucleated mass of protoplasm called plasmodium. The plasmodium consists of diploid nuclei, lacks cell wall, and secretes lime. The plasmodium shows amoeboid movement by producing pseudopodia. Chlorophyll is lacking, so these are generally saprophytic, rarely parasitic, causing abnormal swellings upon the body of hosts. They reproduce by means of spores produced in sporangia. Their spores have cellulosic cell wall and are produced through meiosis. The slime molds are generally allocarpic. The spores germinate to produce myxamebia, or biflagellated swarm cells behaving as gametes. The swarm cells fuse in pairs, showing isogamous types of cell reproduction. Slime molds may be colorless or variously colorless, colored like yellow, orange, brown, etc. Slime molds are of two types, acellular and cellular. Acellular slime molds have a wall-less multinuclear protoplasm or plasmodium without protoplasmodium, or with a protoplasmodium, or with a number of branched veins showing cyclosis. Phaneroplasmodium multiplication occurs by plasmatomy. Normally, when food is about to exhaust, the plasmodium comes to rest and develops sporangium. Each sporangium has a non-cellulosic covering called caprolicium, example Fuligo and Physarum. Cellular slime molds are initially in the form of haploid uninucleate wall-less myxoamoeba, which is surrounded by mucilage. Move about by pseudopodia and feed on bacteria and other microorganisms through ingestion. Myxamoeba multiply by binary fission. Sexual reproduction occurs occasionally through macrocyst formation, example Dictyostelium, polyspondylium. Protozoa, also included in the animal kingdom, are unicellular organisms having varied form structure and holozoic saprobic or parasitic nutrition. Reserve food is glycogen. Cis occurs during unfavorable condition, asexual reproduction by fission or budding, and sexual reproduction by conjugation or syngamy. Major groups of protozoans based on locomotory organs are zooflagellata, flagellated protozoans, sarcopenia amoeboid protozoans, ciliated protozoans, and sporozoa. Okay, now let's go on to some questions on kingdom Monera and later on to kingdom Protista. Right, let's start up with some questions about the kingdoms of Monera and Protista. What are the main differences between Monera and Protista? The main difference between Monera and Protista is that Monera is unicellular and prokaryotic, while Protista is unicellular and eukaryotic. Monera lack well-defined organelles and nuclei, while Protista have well-defined organelles and nuclei. What are some examples of Monera? Some examples of Monera include Bacillus, Halobacterium, Mycobacterium, Sporo, Halobacter, and Clostridium. How does Monera reproduce? They reproduce asexually through binary fission or sexually through conjugation. In unfavorable conditions, they form endospores. Where are Monerans found? They are commonly found in hot springs, snow, and deep oceans. The first eukaryotic cell to evolve would be Protista is the first eukaryotic cell to evolve. What is the role of Monera in our digestive system? Monera break down dead material and food in our digestive systems. Why are protists more developed than Monera? Protista are more developed than Monera because they possess more complex characteristics. Protista are eukaryotes, meaning they have a true nucleus, whereas Monera are prokaryotes, meaning they lack a nucleus. What is the mode of nutrition Monera? The mode of nutrition of all organisms belong to kingdom Monera is autotrophic. Why do protists and Monera have, what do they have in common, protists and Monera? Their similarity between Monerans and protists is the presence of cell membranes. Both Monera and protists have membranes without a cell wall, for example, mycoplasma and antimonera and Euglenoids under protists. What is the economic importance of kingdom Monera? Monerans are very useful organisms. They enrich the soil and serve as an important part of the nitrogen cycle. They also help in the production of some food items and antibiotics Methanogens play an important role in the treatment of sewage. What is the link between Monera and Protists? The correct answer is step-by-step. An imaging solution, dinoflagellates are considered as connecting link. Text and image solution for dinoflagellates are considered as connecting link between Monera and Protista because, as per experts, doubts and scoring. There's some additions on the work there, and I think we can go back. We don't need to connect that. What is a reserve food in Monera? Yes, in case of organisms such as cyanobacteria belonging to the kingdom Monera, the reserve food is in the form of oil and fat droplets and proteinaceous granules. In case of protists such as algae, the food reserve is starch, which is stored in pyrenoids. Monerians are present in both aerobic and anaerobic environments. Some have rigid cell walls, while some do not. So we're just about finishing. What's the source of energy for Monera? It's photosynthesis like purple sulfur bacteria or chemosynthesis